All right, guys, we are on chapter 27 on prokaryotes. And this slide is here just to remind you that the two prokaryotic domains of life are bacteria and archaea. Okay, please recall that the two prokaryotic domains are bacteria and archaea. This is a picture from your textbook. It's just a simplified picture that is reminding you that domain bacteria, archaea, and eukarya all derived from a common ancestor. Now, I'm going to start out just telling you a little bit about archaea and some differences between archaea and bacteria. Okay, so remember, they're both, actually, let me get to a different picture first. Okay, so here is a cartoon of a prokaryotic cell. Okay, and you hopefully you might remember this from Bio 1. Okay, so remember, a prokaryotic cell is a single cell with no compartments, unicellular. You have a circular chromosome, okay, that's made up of double-stranded DNA. Okay, that's common to all of life. Okay, so both bacteria, archaea, and eukarya have chromosome. In bacteria and archaea, it is a circle. Okay, so think of a rubber band and then you twist it up and then it starts to look like spaghetti into knots. Okay, that's what it looks like. Of course, it's within an aqueous environment, which is the cytoplasm. You have ribosomes. Okay, remember, all of life has ribosomes, and it's the ribosomal RNA that was sequenced to identify domain archaea. Okay, you have carbohydrates, ions, enzymes, everything in the cytoplasm. Then you have a cell membrane. Okay, it's not showing this on this picture, but you have a cell membrane, okay, that is basically the container. Remember, cell membranes made of phospholipids. Then outside the cell membrane, okay, you have another layer which is called the cell wall. And you're familiar with the cell wall in plants being composed of cellulose. Okay. Prokaryotes also have cell walls. Okay, I'll tell you their composition in a minute. Okay, the cell walls are made of carbohydrates, so different from cell membranes. Remember, you never mix up cell wall and cell membrane. Okay. Then I sometimes struggle with textbook pictures, but they're showing another layer outside the cell wall, which is called a capsule, and not all prokaryotes have a capsule. So that's why I put that it's optional. Okay. I'm not going to ask you anything about the capsule. Some prokaryotes have a flagella, which allows it to move, and that is a little different from eukaryotic flagella. But again, not all prokaryotes have a flagella. Not all are motile. So that is something that is optional. Okay. Asexpilous, which I will mention a little bit later in this lecture. Some prokaryotes have them, some don't. Okay, so that again is optional. But I do want you to know prokaryotes have a circular chromosome, ribosomes, cytoplasm, cell membrane, and cell wall. Okay, those are the basics you need to know. All right, now let's contrast archaea and bacteria. Bacteria have a carbohydrate called peptidoglycan. Okay, a carbohydrate called peptidoglycan in their cell walls. You see the word glycan, that means sugars. Okay, so it's reminding you it's carbohydrates. Okay. Archaea do not have that same material in their cell walls. Instead, they have a molecule called pseudomurine. Okay, so they are structurally a bit different. Okay, so keep that in mind. Archaea and bacteria are prokaryotes. They have cell walls, but they're a little different. Now, bacteria and eukarya membranes are very, very similar. Okay, Bacteria and eukarya both have cell membranes made up of phospholipids. Okay? Very, very similar. Archaeal membranes are a little bit different. Okay? Their lipids are structurally different than the lipids in bacteria and eukarya. So I want you to understand that not all prokaryotes are the same. Okay, you do have those two different domains with some differences. Now, archaea and bacteria, structurally, like I just went over that picture, right? Single cell, no compartments, right? Cell wall. In that way, archaea and bacteria are very similar. Okay. Now, I'm saying metabolically similar, but I haven't covered that yet. Okay. Now, transcription and translation, that's from Bio 1. Okay. So remember enzymes like RNA polymerase. Okay. It's going to be more similar. RNA polymerase is going to be more similar in archaea and eukarya than archaea and bacteria. Okay, so the enzymes that are involved in DNA genetics, archaea and eukarya are more similar. Bacteria is a little different from eukarya and archaea. Okay. So an example would be eukarya needs more transcription factors in addition to RNA polymerase than bacteria, and so does archaea. Okay. I'm telling you these things so you keep in mind they are 3 different domains. You don't have to memorize this picture, okay? But just note that they're showing that the bacterial or eukaryotic lipid, so they're the same, okay? If you recall from Bio 1, you have your 3-carbon glycerol, and you have 2 fatty acids, and of course there would be a phosphate here. Okay. Now structurally, the archaeal lipid chemically is a little different. Okay. And also, in bacteria and eukarya, you always have a phospholipid bilayer, right? You have 2 layers of phospholipids. In archaea, there's a possibility of having a structure that is a monolayer. Okay. So there are some differences there. Now, for prokaryotes. Now, when I'm saying prokaryotes, I'm referring to both archaea and bacteria. They are ubiquitous. The word, and I don't have it written here, ubiquitous, ubiquitous. That means they are found everywhere. Okay, they're everywhere in the environment. You're going to find them in really hot environments, really cold environments, and medium temperature environments, right? So thermal hot springs, you will find them in the Arctic, no problem, okay? You will find them in very acidic, very basic, or very neutral environments. You'll find them in fresh water and salt water, and then you can find them in like really concentrated salt environments as well. Okay. So they're found everywhere, every environment you can imagine. Now, if they are to find life on Mars, okay, you know how there's always talking about finding life on Mars? The life that they're going to find is prokaryotes, first and foremost. If you find prokaryotes, then you know that you have an environment that is habitable. Okay. So when people are looking for life on Mars, they're mostly looking for things like prokaryotes right now. Now, prokaryotes are microscopic. Okay, bacteria and archaea, they're a single cell, so you'll never see them by the naked eye. Okay, you have to use a microscope. But there's loads of them everywhere. Okay. So, within just like a handful of soil, you know, you're going to get billions of prokaryotes and of a wide variety of different species. Okay. So, they're abundant everywhere, and just be aware of that. Okay. I've already gone over this picture. All right, now let's talk about size. All right, so if you get a ruler, okay, you know what a centimeter looks like on a ruler. And well, you did online last semester. But, you know, on a ruler, you see the centimeter mark. Okay, between centimeter 1 and centimeter 2, you see 10 tick marks. If you want to go get pause and go get a ruler, okay, or just Google a ruler. And then with one of those tick marks is a millimeter. Okay, and you know you can see a millimeter by the naked eye. Okay, right, and 10 millimeters is a centimeter. You know you can see that by the naked eye. A millimeter, that you can also see by the naked eye. Now there's this unit. This means micrometer, okay? That is a Greek M, okay? That's micrometer, the symbol. If you take one of those millimeters and you divide it by 1,000, okay? Divide it up in 1,000, then you end up with what is a micrometer, okay? So there are 1,000 micrometers in 1 millimeter. Prokaryotic and eukaryotic cells are in the micrometer range. Okay, eukaryotic cells are larger micrometer, okay, averaging about 100 micrometers. And this is showing a yeast cell, and you can see them quite easily in the microscope. Okay, and yeah, it's too bad you didn't do in-person lab, and then you would have seen that. Okay. Now, when we go down to prokaryotes, here are 2 examples of prokaryotic cells. A bacterium in the genus Bacillus and a bacterium in the genus Escherichia coli, in the genus Escherichia. Okay. When you hear E. coli, this is what they're referring to. And they are all in the micrometer range, okay, about 1 micrometer to about 5 micrometers. And for them, you need to use a light microscope to visualize, and usually you have to magnify 1,000 times to really get a good image. Okay. The next unit that gets used is nanometer. Okay, so if you take one of those millimeters on the ruler and you divide it by 1,000,000, then you'll get a nanometer. Okay, and things that are nanometer sized are things like viruses. Okay, right? Like SARS-CoV-2, anything like a ribosome, proteins, all of those are nanometer. And you can't see them with a light microscope, so you have to use a fancy electron microscope. But I want you to know prokaryotes are in this range, about a micrometer, roughly, not visible to the naked eye. Okay, you have to use a microscope for those. Prokaryotes have since it's a single cell, it has a minimal variety of shapes. Okay, one shape is called bacillus. Okay, bacillus, usually it's with a lowercase b. Bacillus, another word for that would be rod shape. Okay, so you see this is a rod or bacillus shaped prokaryote. The other shape it usually comes in is coccus. Another way you can think of coccus is spherical. Okay, so bacteria in the genus Bacillus are going to be rod-shaped. Okay. Think of the bacterium Staphylococcus. Okay, that tells you it's going to be these round spherical-shaped bacteria. Some bacteria have a little more shape to them, and they are called spirillum. So kind of think permed hair. There's some curve in it. So it's like a curved rod. That's spirillum, a spiral. Those are the most common shapes. So when you compare that to eukaryotes, right, where you have multicellular organisms and you can come up with a lot more shapes, pretty minimal with prokaryotes. Okay, so their diversity does not come into what they look like, because they're a single cell with, very few shape variants. So I've already gone over that, but the first living organisms that evolved are prokaryotes. Okay, remember the prokaryotes are closer to the root of the tree of life, okay? And we know that. Your prokaryotes arose before your eukaryotes. I should edit this. It shouldn't say most, because pretty much prokaryotes are unicellular. Okay, so prokaryotes are unicellular. Okay, I've already gone over the size, and I've already gone over the main shapes. Now, prokaryotic cells has a variety of shapes. Yeah, I should have probably edited that part out too, because they don't have that huge of a variety. I mean, I could come up with some other varieties that I'm not going to cover because this is an intro class. So what we're doing right now in this course is doing a survey, okay, of life. To go into more depth on prokaryotes, you would have to take a microbiology class. Okay, so this is a survey. We're just doing an introduction to prokaryotes. So I'm going to pause for a second. Okay, before I go on to this slide, what you're going to want to have, hopefully you've already printed out your study guide, right? Your Unit 1 study guide, okay? So I think it's page 5, but I'm not 100%. Okay, go to page 5, and what you're going to see are tables, okay? And they're tables for you to describe characteristics of bacteria, archaea, and then we will get to protists underneath. And I want you to notice the type of things I put in each row that I want you to describe. So for basically what you're going to want to do is think of a few parameters, okay, and describe organisms based on these parameters so you can really compare and contrast. Okay, so just for example, you see characteristic, prokaryotic or eukaryotic, unicellular, multicellular, both, right? Ribosome size, cell wall presence, composition, cell membrane presence, composition, all these kind of things. And you will find all of these things will pretty much be addressed in these lectures, okay? Yes. Okay, now I'm just noticing one thing I may not have clearly addressed. Well, some of the, just, you gotta hang on. I'll get to all of this. Okay, and if I don't, just email me and ask me. Okay. All right, so we did the basic structural stuff. Okay, but one structural thing that I did not mention is the ribosome size. Okay, so the ribosome size in prokaryotes is 7ds. Okay. In eukaryotes, the ribosome size is 80s. And you remember the ribosome is responsible for protein synthesis. Okay. And just know the eukaryotic ribosome is a little bigger. Okay. It's 80s, whereas the prokaryotic bacteria and archaea are 70s. Okay. Now, let's talk about some benefits of prokaryotes, okay? So people often think of things like bacteria as germs and as bad, okay? But they're not, okay? The vast majority of prokaryotes are beneficial. They're beneficial directly to you and to the environment, okay? So the NIH might be almost 2 decades now, I'm not 100% sure the date, launched the National Institute of Health Human Microbiome Project. And they didn't do it alone. It was in combination with labs around the world to catalog all the microbes associated with the human body. Okay. And it turns out there are more bacteria associated with the human body than there are human cells. Okay, so an adult human has trillions of cells, okay, or a human has trillions of cells. And there's approximately 10 times more bacterial cells associated with the human. So often a human is considered a superorganism because it's the result of the activity of the human cells as well as the bacterial cells. It's easy to imagine that there's 10 times as many because they're so much smaller, you could just fit a lot more On one eukaryotic cell, you could easily fit hundreds of bacteria. Okay. Now, I'm focusing on bacteria because bacteria associate with humans far more than archaea does. Okay. Archaea are primarily environmental organisms that play a role in the environment, which I will be telling you about momentarily. But there are some archaea in the human gut. But mostly associated with humans is bacteria and also some yeasts. Where in a human you find bacteria is in the nasal passages, in the mouth. The skin is covered in bacteria along with some yeasts. Where the vast bulk of the bacteria associated with the human is in the gut. Okay, so gastrointestinal tract. Gastro means stomach, intestinal means small and large intestine, and often we're referring to the intestines as the gut, and really most of the bacteria associated with the human is in the large intestine. Okay, and then there is some in the urogenital area. Okay. Now there has been quite a few studies of gut bacteria, and it has been found that gut bacteria play a role in human health. They play a role in digestion, okay? And it has been shown that what you eat will alter your gut bacteria, which can alter your metabolism, okay? So if you use NPR or New York Times, they do a lot of studies on human microbiome. You may have heard of probiotics, okay? That's referring to maintaining the normal gut bacteria. Okay, so we know they play a big role in human health. Okay, and so most are good. They help you. Okay, but how they help you? So we call them normal bacteria flora. One of the things they do is help with digestion. Okay. We know some of the gut bacteria produce vitamins, and they also produce compounds that are anti-inflammatory, okay? And it's inflammatory compounds that are going to lead to things like IBS, irritable bowel syndrome, okay? So anti-inflammatory compounds are going to calm that down. But really the most important role that bacteria flora play is preventing growth of pathogens. So bacteria are going to be able to stick to cells, let's say skin cells, cheek cells, right, respiratory cells, your gut cells, and if they're sticking and they're taking up nutrients and benefiting the host, then there's no space for the pathogen to take up residence. Okay? If they're already sitting at the table, they've taken up that house, they're eating the resources, then there's no space for a pathogen. Okay. So if you sometimes take antibiotics, and what antibiotics do is inhibit the growth of bacteria, and it's when you take antibiotics, it's because you've had bacteria that have overgrown, okay? And the antibiotics inhibit their growth, but in the alongside, the antibiotics are going to also stop the growth of other beneficial microbes, okay? Because they're not really good at specifically stopping the growth of one bacterium versus another because you've got so many. And so if you ever get wonky gut from taking antibiotics, like your gut, you get kind of diarrhea, that is because you've lost, at least temporarily, some of your gut bacteria, right? And so then there's an intimate homeostasis balance between all the microbes that associate with you. Okay, they communicate with your cells. Once you've lost that homeostasis is off, and you start to feel icky. Okay, so just keep that in mind. Okay, we do have bacterial flora with us, and they benefit us. Okay, and one of the ways they benefit us is by preventing pathogens, okay, from being able to grow up to large numbers and cause disease because they use up the resources in the space. If you have more questions and you want to know more about this, feel free to ask me. I can only go so far, okay, in an intro course. Other really important activities of prokaryotes, and I use this slide in my micro class, that's why I use microbes here, but let's just say prokaryotes. Okay. The first prokaryotes on Earth were called cyanobacteria, 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 and they do photosynthesis. Okay, so remember, it's cyanobacteria that are critical for the creation of the oxygenic environment. And there are many prokaryotes that currently do photosynthesis and produce oxygen. You're going to find those prokaryotes in aquatic systems, okay, in freshwater and marine systems. So it's not only the plants that produce oxygen for the environment, prokaryotes do too. Okay. Nutrient cycling and decomposition. Okay. Prokaryotes do a very wide range of chemistries. Okay. They do a really wide range of chemistries, and so they make enzymes that are really good at breaking things down. Okay. So, and they decompose things, right? They break them down. And so that's how you get food spoilage, right? Food, when it breaks down, it starts to get kind of liquidy, right? That decomposition occurs from prokaryotes. Okay. And when you get decomposition, then those larger chemical compounds are getting broken down to the elements, right, that can then be used to grow another organism. So that's the nutrient cycling. Something that has died gets broken down, and those nutrients and elements get used to build up something else. Okay. Prokaryotes play, and all the bacteria associated with the human play a big role in decomposition of humans and mammals. Okay. So like once a body dies, right? And you always hear about like floating bodies, right? If you don't weigh it down enough, or it gets loose and it comes up, it's the gut bacteria that are going to start decomposing, right? Feeding off of that dead carcass. And in the process, the chemistry, they're going to produce a bunch of gas, right? So then the intestine will bloat, right? Fill up with gas and then air, and then it'll float up. Okay. So cellulose, which is the cell wall of plants, okay? Animals, we do not have enzymes. Well, most of us. Cows do, but like humans do not have the enzymes to break cellulose down, but a lot of prokaryotes do. Okay? So they can break down plant matter in a different way than humans can. Bioremediation, remedy here means remedy, to remedy. So bio, to remedy with bio, okay, with biological substances. Prokaryotes do a variety of chemistry, so they can be used in bioremediation and waste treatment to remove pollutants. Okay, so they're used in like the BP oil spill, in polluted sites. You can add them and they'll pull out that pollutant from the environment. I'm not going to go, so it's usually yeasts that are used for making bread and cheese, but for yogurt, okay, for yogurt, it's a prokaryote bacteria called Lactobacillus and Streptococcus. So if you go look at a yogurt container, you're going to see Lactobacillus and Streptococcus, and those are prokaryotes. Okay, and you'll often hear that if your gut feels inky, then go have some yogurt, and that's going to basically function as a probiotic. So you'll repopulate your gut with good, beneficial bacteria. A lot of antibiotics and drugs, pharmaceuticals, use prokaryotes. Okay? So for example, E. coli is a bacterium and we can manipulate the genetics of bacteria quite easily. Okay? And you can put in different genes, and so and then ask the prokaryote to pump out that gene product. Okay, so for example, when insulin was first getting manufactured, it was taking, I think, pig pancreas, okay, and isolating it from pig tissue. Okay. You can imagine that's quite laborious to get pig tissue, grind up, and extract that insulin. Okay. And it's not necessarily very cost effective. Figured out putting the insulin gene in an E. coli and asking the E. coli to do transcription and translation and make that insulin protein and then harvest it. Okay. The source of many of our antibiotics are bacteria. Soil bacteria make a lot of antibiotics. They don't make enough in the soil that you can extract it from soil, but you can isolate those bacteria and then grow them in the laboratory, put them in the conditions where they make a lot of those antibiotics, and then harvest them. I'm not going to go into ethanol and biofuel much, just because I think I've given you enough things to show you the importance of microbes. They play of prokaryotes. They play a role in human and animal health, like in the gut, and then all of these variety of things like oxygen production, nutrient cycling, bioremediation, drug production, okay, food production. All right, so there's a book called the Burgess Manual of Determinative Bacteriology, and in there are categorized about 5,000 bacterial species that have been characterized in the lab. They've been characterized structurally and chemically. Okay. Now, that's probably not all of the bacteria in nature. There are probably plenty of other bacteria in nature that we don't know about or haven't characterized because we haven't figured out how to grow them in the laboratory. Okay. So, by no means is 5,000 the max number of prokaryotes. There are more out there. We just haven't necessarily gotten to the point where we can fully characterize them. But only a small subset, about 200 of them, actually cause disease. Okay, most of them are completely harmless. Okay, and if you want to know more about bacteria, ask me. I'm happy to talk about it. That is actually my specialty. Okay, or take a micro course. Okay, now let's go into some more nitty-gritty, and I'm not going to talk about really archaea much anymore. I basically contrasted archaea and bacteria for you already. Okay. Now, both archaea, well, I'll mention them both back and forth. Okay, both archaea and bacteria have a cell wall. Okay, and the function of the cell wall is to maintain the cell shape and prevent the cells from bursting in a hypotonic environment. You may recall from Bio 1 that if you put plant cells in a hypotonic environment, right, like in a dilute, like let's say fresh water, the cells don't burst. Okay, water will rush in, they'll swell, but they don't burst in plants. That's because they have a rigid cell wall that maintains the shape. That's the same thing for bacteria and archaea. In bacteria, the cell wall is made up of peptidoglycan. Okay, peptidoglycan, and that is a unique polysaccharide only found in bacteria. Okay. Now, antibiotics stop the growth of bacteria, and many antibiotics target the production of peptidoglycan. It targets the production of peptidoglycan, then it stops the bacterial cell wall from being made. So then the bacteria cells are more easily burst. Okay? And then if they're burst, they can't reproduce. Okay? So just know that peptidoglycan is one of the things that is targeted by antibacterial drugs. Now, your textbook goes into more detail about this, and we can subcharacterize bacteria based on their types of cell walls. Okay? They have two predominant types of cell walls, and one is called a gram-positive cell wall, and the other is called a gram-negative cell wall. And there was a scientist named Graham who basically figured this out but basically by playing with dyes. Okay, taking cells, playing with dyes, and asking which cells pick up which color dye and which ones don't. Okay, and if he had found cells that would pick up dyes differently, then that means they were chemically different. Okay, so he knew that when he took cells and he colored them with dye, some would, I don't know how easy it is to tell on this picture, but some would be purple and some would be pink. Okay, so then the ones that ended up getting colored pink, he called gram-negative, and the ones that got colored purple, he called gram-positive. But he didn't know why. And later we found out why, and it is because their cell wall arrangements are different. Now, for a survey class like this, I do not feel like it is important for you to memorize the differences between a gram-positive and gram-negative cell wall. So I'm not going over that. Okay, you're free to ask me about it, but if you take a microbiology course, you'll definitely get that. But the reason that we characterize organisms as gram-positive and gram-negative is by getting information about their cell walls, then we know how to characterize and treat those bacterial infections. Okay, it's just more information. Okay, now, how do prokaryotes divide? Actually, what I mean by divide, another thing is reproduction. Okay, so I think your chart is going to mention mode of reproduction. The mode of reproduction is called binary fission. Okay, this is for archaea and bacteria. They reproduce by binary fission. And binary fission is a lot like mitosis, in that you start out with one cell, you double all the contents, you sort all the contents, and you end up with two daughter cells that are identical to each other and to the parent cell. Okay. But the reason we don't call it mitosis is because prokaryotes do not have a cytoskeleton the way eukaryotes do. And there's no nucleus. Okay? So you're not dealing with microtubules or nuclear membranes or any of those things. So since you're using different structures in the cell, it's not the same process. Okay. So prokaryotes divide by binary fission. Okay? And what they're showing here is the chromosome gets copied. That's DNA replication. So you get 2 copies of the chromosome. I don't know why this isn't from your book. I don't know why it colored them differently, because it makes it seem they're different, but they're not. They're exact copies. Okay. Notice there's ribosomes inside the cell, right? Cell membrane, cell wall. Okay. Then there's what's formed a septum, okay, which is basically a kind of bridge, indentation marking the exact middle of the cell. Okay. Now notice the cell has gotten bigger, right? It's doubled, okay, because it's doubled all of its contents. So it's going to get bigger if it doubles. Then you get this symmetrical septum, okay. On either side will go one chromosome, will go the other, and then a variety of bacterial proteins will play a role in actually pinching and splitting off these 2 cells. So you end up with 2 daughter cells that are exactly the same as each other, as well as the parent. And that is binary fission, and that's how prokaryotes reproduce. 1 to 2, 2 to 4, and so on and so on. And they do this very, very fast. Okay. For bacteria like E. coli and Staphylococcus, this will happen in about 30 minutes. Okay. Really, really fast. You don't have to memorize all these numbers, and you don't even have to know this word, exponential growth, okay? But it's showing one cell will double, okay, into 2 by binary fission in 30 minutes. The next 30 minutes, each of these will divide into 2, so 2 to 4. After another 30 minutes, each of these will go to 2. So that would be meaning 8, okay? Now, if you were to chart it out, after 10 hours of growth, okay, because every cell divides into 2, after 10 hours of growth, one cell will give you up to 1 million cells. Okay, that's really fast. Okay, that's how prokaryotes can grow. So prokaryotes grow much faster, okay, than eukaryotic cells. One of the reasons is they're really small, okay? So there's a really high surface area to volume ratio, okay? There's a lot of surface because they're really small, and that means there's a lot of place for exchange of material. So nutrients get in quick, chemistry happens fast, wastes go out quick. So what this is kind of showing is if you have bacterial cells from any environment, okay, and then you put them on an agar petri dish. You guys might have some familiarity with these agar petri dishes, okay? And if you spread them out, one of these yellow bacteria would land in a place. One of these reddish ones would land in a place, okay? And then even if you have 1 or 2 cells that land, then they go through binary fission. Binary fission, many rounds of binary fission, okay? Until there's 1 million or 1 billion cells piled up on top of each other. And then you can see those 1 million or 1 billion cells as what's called a colony. Okay, so these are showing these petri dishes with these different colonies of bacteria. Okay, so but this red colony would be made up of millions or billions of microscopic red bacteria. The yellow one, millions or billions of these yellow microscopic cells piled on each other. This white one, that would be millions or billions of that. Most bacterial colonies are going to be like cream or off-white. Okay? Most aren't going to be red. You'll get some yellow. So that's what these are showing. Okay, and you can take a petri dish, put it in pretty much any environment, swab any environment, and put it on a plate, and you will see bacteria growing. Now, what some people do, there are some bacteria that are, of course, red, some yellow, not many, some purple, and some people will use the bacteria and then, like, toothpicks, and then draw, basically do bacterial art. And there's contests for the prettiest bacterial art pictures. So I guess you could even just Google, petri dish art, right? Or bacteria art, and you would find a fair amount of cool stuff. Okay. But the only reason these colonies are visible is because there's millions of them piled on each other. Okay, so prokaryotes reproduce by binary fission. So the offspring cells, the daughter progeny cells, are genetically identical to each other and the parents. Now, mutation in living organisms is low, right? DNA polymerase that does DNA replication is really good at editing. Okay, it might have been a while since you've had this, but just know DNA polymerase does DNA replication, and it does a really good job of editing, so you don't get a lot of mutations. Okay, and DNA polymerase and replication happens the same across all of life, basically across bacteria, archaea, and eukarya, and you get low mutation rates. Okay, that's why it takes usually quite some years, and you're older, for cancer to show up. Okay, because it takes time to build up those mutations, because mutation is rare in living organisms. In contrast, there's different variants of coronavirus, of SARS-CoV-2 that leads to COVID. Viruses mutate at a much faster rate than living organisms. So viruses are not considered living, okay? They don't have the same editing DNA polymerase. Okay, so they mutate much, much faster. But because bacteria reproduce so fast, they go through one generation, one parent to daughter, in 30 minutes. You can go to granddaughter in an hour, right? Great-granddaughter in an hour and a half. You go so fast that those mutations arise very quickly, okay? Because it's all about generation time. Okay? So you can actually see evolution happening in real time with prokaryotes, okay? by growing them in the laboratory because you can get them through millions of generations in a short period of time. An example of an evolving bacterial population is antibiotic-resistant bacteria. You may have heard of antibiotic-resistant bacteria, ones that aren't killed by antibiotics. They derived from bacteria that were sensitive to the antibiotic, could get killed, and then it picked up mutations over time that made it resist that antibiotic. So that was evolution, and that happens in real time. Okay, the chemistries, of what prokaryotes do. That's really where the difference is among prokaryotes occur. It doesn't really come again what they look like, because they're only one cell. Okay, their shape, they're really only three. There are three main shape differences: rod, coccus, or spiral. There's only two different kinds of cell wall types, mainly gram-negative and gram-positive. Okay, so that's not a lot of variation. Where the variation in prokaryotes comes in is their chemistry. What kind of chemistry do they use to make ATP? Okay, if you recall, it's catabolic reactions that allow for the generation of ATP. Hopefully you guys remember that animals do aerobic cellular respiration, right? Where they take organic compounds, break them down, along with O2, and then make ATP, CO2, and water. Okay. So many prokaryotes use oxygen just like we do and perform aerobic cellular respiration. Okay. So some really require O2 and use that O2 to perform aerobic cellular respiration as their main mode of ATP generation. Some prokaryotes are poisoned by O2, so they have different O2 requirements. All animals require O2, okay? But some prokaryotes are poisoned by O2. And they only do anaerobic metabolism. Anaerobic meaning and without oxygen. The two types of anaerobic metabolism, if you recall, one is fermentation, okay? So basically the same fermentation you learned about in Bio 1, and a process that we haven't covered, which is called anaerobic respiration. Some prokaryotes can use O2 or not use O2. They're very flexible. They can survive or grow with or without O2. So then they can do aerobic cellular respiration, anaerobic cellular respiration, and fermentation. Okay. So in prokaryotes, you have three mechanisms of ATP production. Okay. Whereas animals are dependent on aerobic cellular respiration. So you have more diverse methods of generating ATP energy with or without O2. All right, next thing to bring up is energy source and carbon sources. Okay, so all organisms have to have an energy source and a source of carbon to be able to generate energy and reproduce. Okay, animals, our carbon source is we eat organic carbon. And we use the same organic carbon as our energy source because we break the bonds in the organic carbon for energy. Plants use CO2, right? Atmospheric carbon dioxide to convert it and then convert it to organic carbon. But they use carbon dioxide as their carbon source and sunlight as their energy source, right? Photosynthesis to make the organic carbon. Prokaryotes, and you don't necessarily have to have all this memorized, but I want you to have the sense that prokaryotes are very diverse, okay? Depending on the prokaryote, they can use organic carbon as their carbon source or inorganic carbon as their carbon source. Okay, organic or inorganic. It's inorganic is CO2. Okay, so whereas animals can only use one, plants can only use one, prokaryotes can use either or. Okay, prokaryotes can do photosynthesis like cyanobacteria, so some will use sunlight. Okay, not all, but some. Some are going to use chemical compounds. Okay, the prokaryotes that associate with humans are going to use chemical compounds. Right? If they're in our gut, there's no light. They're not doing photosynthesis. Now, the chemical compounds they can use organic chemicals, right? Sugars, amino acids, proteins. Or they can use inorganic chemicals, right? Not carbon-based. Okay. So just see the variety. Sunlight, organic chemical, inorganic chemicals. Okay. So great diversity in their carbon sources and energy sources. Okay. Now, if an organism uses light as an energy source and carbon dioxide as a carbon source, we call it a photoautotroph. You're going to see the word troph a lot. Troph means feeding. How does it feed? Okay, it's going to feed off of sunlight, photo, and the term midfix auto is used for CO2. Okay, so both plants and prokaryotes are photoautotrophs. Organisms that use organic compounds as their energy and carbon source, I just ran out of space here. Okay, those would be called let's just say chemoheterotrophs. Okay, or chemoorgano. So chemo, organo. Okay, organic chemical. Hetero meaning, again, a variety of organic chemicals. Chemoorganoheterotroph. That's what we are. Okay. You can also just abbreviate it to chemoorganotroph. You don't have to have that memorized, by the way, at all. Now, this is the unique thing I want you to know about prokaryotes. So prokaryotes in animals can use organic compounds as energy and carbon source and can be chemotrophs. Can use sunlight and carbon dioxide and be phototrophs. Okay, so notice I'm abbreviating this to phototroph. I'm abbreviating this to chemotroph. Okay. But this is something unique, okay, that animals cannot do and plants cannot do. And this is called rock eating. Okay. These inorganic chemicals are found in rocks. Okay. So the organisms that use these as their main energy source would be called rock eaters. Okay, so these would be organisms that use inorganic chemicals as energy and carbon dioxide, so no organics at all. And those would be called chemolithotrophs. Okay, so that's a new category of metabolism that prokaryotes do that animals and plants can't. Okay, rock eating, which is called chemolithotrophy. So basically just want you to know they are very diverse in their chemistries, in their carbon source, energy source, and oxygen requirement. Okay. So they're not going to look a lot very different. The chemistries and the metabolism they can do varies quite a bit. And so they're really cool in that way. All right, and I am probably going to stop right here because we're at 54 minutes. And I'll continue the rest in the next video.